Greetings to everyone in the research phase or of a newly enrolled breed. Today I want to talk about how we select our initial sample of dogs when we are assessing a breed for genetic diversity. This can help us know whether or not a breed is well distributed in the genetic diversity that is at the breed-wide level, which we call biodiversity or allelic richness. We can also determine whether or not there's a genetic bottleneck within a breed, as long as we have selected a diverse and unrelated initial sample as possible. So why is that important? Why is it important to get as diverse an initial sample as possible? Let's say that this was your initial sample. When we look at this sample, it looks like there's two significant outlier dogs, the blue one over here and this red one over here, while the other dogs in the population look like they're more closely related. But what if this was the actual population of dogs, and we had a whole subset of dogs that were not sampled over here, plus another dog over here? Or what if this is the actual population? Back to the original sample, we would think that the outlier dogs are relatively unrelated to the majority of other dogs in this population, and relatively unrelated to one another. Now these dog images I'm making, which I did not say earlier, are meant to look like the PCOA graphs, which is the visualizations, 3D visualization of how a population relates to one another. And you'll see them in your breed reports. And so basically where the dogs cluster kind of can indicate to you how closely related they are to one another. And then when they're farther apart, we would consider them relatively unrelated in relation to the rest of the population. Okay, so when we look at this original one, we think that the outlier dogs are relatively unrelated to one another and the rest of the population. But then in the second scenario, we see that only one of the dogs we thought was genetically different from the gene pool actually is atypical. The two originally thought to be outlier dogs are also more closely related to one another than we originally thought. So if you look at here, they seem closer together now than, say, this dog all the way to this dog. They're closer related. So it's important that we sampled correctly. Okay, and then in this third scenario, none of the initial sample are actually unique for the actual population, and they are much closer, more closely related than we thought they were. So you look here, they all look a lot closer together once you're looking at the whole actual population, assuming that our initial sample was not a good one. So the way our algorithms look to predict how closely related dogs are to one another is based on our population. So we can only know what we know about a population. And our algorithms predict relatedness based on the population that's been tested. If the initial sample does not test the breadth of the genetic diversity of a population, the results will drastically change over time, depending on how good our initial sample was. So let's say in this sample here, all these pink dogs are all from the same family, while all the rest of the sample are unrelated via pedigree. Overrepresenting a line of dogs in the initial sample will make them seem more typical than they might actually be uncommon in relation to the rest of the population. If we tested only one member of that family in the original sample, you would see that they're actually very atypical or less typical in relation to other dogs that were carefully selected. So say none of the rest of these are actually siblings or related. You can see that that family is actually very atypical. Alternatively, let's say this dark green dog at the bottom here is actually really common in a region of the world, or say it's a particular coat type or color, or it's a performance dog, and we did not do a very good job sampling that. It'll make it seem like it's very atypical. Alternatively, if we say that dark green dog on the bottom is actually really common for a region of the world, or it's for a performance dog, or for a specific color or function, but there's only one that's been tested, it'll make it seem like it's atypical when it's really actually very typical. Our initial sample should sample the depth and breadth of genetics worldwide as best as possible. So what way would your breed be split? We recommend thinking about your breed by several different factors. So what all the different countries of the world that the dogs exist in, the different variety of pedigrees. Are there atypical pedigrees that have less commonly represented dogs? Does your breed have a color of coat or coat type that's different and might have split your breed into different distinguishable lines? Or is there potentially a difference in purpose, so conformation, performance, or pet? There's lots of different things and variations like this in each breed population. So you want to divide up those factors to determine how to sample your breed. If there is a divide between show, performance, and pet lines or location, we want to make sure that they're all sampled. We may not find that there is a genetic difference, but it's important to cast the net as wide as possible in the initial sample to get the best initial sample for our calculations. Now remember, we're sampling as unrelated as possible within a breed. All breeds will have relatives at some point in the pedigree because that is how breeds are formed. If we do a good job with our initial sample, we will have a powerful tool to help manage your breed's future. Let me know if you have any questions, and thanks for watching.